Welcome back to the course on functional genomics. So, in the previous lectures we have seen how one could engineer the genome and look into the function of a variety of genes in terms of a given process. We have looked at the example of cell division mitosis, we have looked at the examples of virus infection process, and then some of the developmental disorders people have modeled using or identified the genes using zebrafish and so on. So, what we are going to do in this lecture and the few lectures that are following this one is to understand what are the tools, contemporary genomics tools people use to understand the expression of the genes. Basically, we are going to look into the genome wide expression studies, right. We look into the history and the tools, how they are developed to understand what are the genes that are expressed. For example, you are looking at two different sources, tissue 1, tissue 2, or it could be a normal condition and disease condition. And what is the difference in terms of expression? When you talk about expression, it is not whether or not a given gene is expressed. We are also looking at the change in the expression level, the amount of the mRNA that is being made for a given gene. So, that also dictates, because higher the protein, lower the protein, it can change the way the cell behaves. Therefore, you need to have methods to even measure the level of expression, meaning the number of transcripts that are being made. So, before we get into, let us look into some of the basic concepts that we are going into what is called as central dogma of biology, the classic view. The classic view is that DNA replicates, makes copies of itself, that is how it is able to divide and sustain. And then there is a process called as a transcription, which makes the RNA. And the RNA is translated into protein. That is what shown here in terms of an eukaryotic cell. You have the nucleus, you have the DNA makes RNA and processed to form messenger RNA, and that is being translated to form the peptide or the functional protein. There are exceptions, probably most of you are aware of the exceptions, what is called as the modern view of central dogma, that is that we discussed in the previous lectures, that you also have a reverse process, what you call as reverse transcriptase, which converts an RNA into DNA, and that there are viruses that exist in RNA form, RNA genome, and there are also proteins that are self-propagating, meaning they can make their own copies and so on. So, there are exceptions, but all these exceptions are rare events. But if you look into the majority of the species, living entities, then you will find that still the original proposal of central dogma biology is valid, and whatever the other exceptions that we said are exceptions. But what is important to know here is that it is not always that the DNA is able to bring about a change in the cell system in the form of a protein via an RNA. In fact, now you know that the majority of our transcripts that are made in our cells, or including humans, function as an RNA. These are called as regulatory RNAs or non-coding RNAs. In fact, this fraction is much larger than the RNA that codes for the protein. So, these are called as non-coding RNA. That is the real major deviation, and as I told you, these are not rare events. Now, people are trying to understand the function of this non-coding RNA. Some example we have seen in the previous lecture, what is called as a microRNA or miRNA, where we have discussed how this microRNA is cleaved into smaller fragments, and these fragments go and bind to some of the target RNAs and destabilize them or prevent their translation and so on. So, one such microRNA can effectively regulate a large number of genes. So, that really shows that these non-coding RNA, which do not really give the protein as a final product and not even helping in the translation process, are able to regulate a variety of processes. So, these are not exceptions, but these are very common, that is what we believe. So, how do we analyze these RNA? That is a big question, because analyzing RNA is again tricky, because you are going to look at the steady state, because the RNA is being continuously made. Some RNA may not be translated, they are made, they are kept there. Some RNA are not making the protein, but function as an non-coding RNA. So, there is a huge challenge with regard to analysis of RNA. And secondly, if you are looking at where the RNA is localized, right. So, you want to look into the localization of the RNAs, that becomes much more challenging. So, let us look into other challenges the RNA pose you. It is not that one gene makes one type of RNA, right, that is not the case. Now, we know that the RNAs undergo what is called a splicing. So, what is shown here is a schematic of a gene which has, for example, introns here and here. When this RNA is processed, the final mature product, the intronic regions are spliced out, and we said that some of these introns can function as an mRNA. These are possibilities, but you have the protein. And of course, we spoke about how the splicing happens and so on. If the splicing does not take place, then you are going to have the introns retained in the RNA, and these introns can introduce some change in the reading frame, because you have a triplet, and if you continue to make triplets, because the introns are present here, the reading frame will be shifted. So, therefore, we are going to have that. But this you will normally call it as an aberration, because the intron should be there and should have been removed, and it is present now, therefore, it is an aberration. But what we know is that such things are happening in the system. So, there are RNAs which have exons like is shown here 1, 2 and 4 different exons, and now how this pre-mRNA having these 4 exons are assembled after the splicing process could be vary. For example, in this particular example, you have exon 1, 2 and 3 joined together, whereas in other it is exon 1, 3 and 4 joined together. As a result, the coding sequence of this transcript and this transcripts are going to be very different, because this region that shown here in red color may have a common coding region, but beyond that the reading frame will be shifted. Therefore, this protein and this protein may look very different and may do very different kind of a function. It is a possibility. So, a same gene which makes one pre-mRNA now can end up making different kinds of protein because of the way the splicing takes place. So, that is called as alternative splicing. So, therefore, it is a huge challenge, and there are a number of studies that are shown that a given gene, depending on which tissue it is expressed, which stage of the development it expressed, it can make a variety of different splice forms, and each one has a different coding potential, and there is a relative level between these transcripts. For example, what is shown here is a tau gene that codes for one of the protein called tau, which is implicated in Alzheimer's condition. So, it undergoes alternative splicing, and it is shown that in aged brain, the way it is spliced, if it is modified, right, the person may end up developing dementia, a process by which you lose whatever you have learned, memory loss and other symptoms that are associated with Alzheimer's disease. So, therefore, it is extremely challenging. So, it is not when you are talking about transcriptional analysis, meaning when you are looking at the messenger RNA expression, it is not simply whether a gene is expressed or not expressed, it is not simply whether how much the transcript is expressed, but it is also which transcript is expressed, because there are a variety of splice variants. So, you cannot go with certain molecular weight, because the same molecular weight RNA can have different splice variants. So, it is a challenging task. So, that is something that has to be addressed. So, if you look into the other challenges, it is not traditionally the messenger RNA. You have what is called as non-coding RNAs, and there are a variety of different types. Some are long, some are short. So, the approach that you are going to use for each one of them are going to be different, and you have to distinguish one group from the other, because you cannot use the same group approach for same group. So, what is known is that you have this, these are some of the classification that has come from a recent paper, which says that you have this protein coding genes, about 20,000 odd, whatever it is. Then you have the other types. These are called as small non-coding RNA genes, because they code for what is called as microRNA and so on, which are very short RNAs. You can see these are microRNAs, other small non-coding regions, RNAs. Then you have what is called as the long non-coding RNA. These are pretty long, because it can run into several bases, And there are other types, for example, intronic regions and others. So, it only shows you the complexity that you face when you want to analyze the transcript. Okay. So, the answer is, do we have all the approaches to identify all the different types of RNA? The answer is no. We are there are, for some of these RNAs, the approach is yet to be fully developed. We do have quite good number of tools to understand the expression levels of protein coding genes. Now we are having even approach to identify the microRNA. But as you discover more and more RNA, because even that is a challenging tool now. So, what are the RNAs that are really the non-coding RNAs, but having a regulatory function? So, that you need to dissect first. So, it is an evolving field, as is the case in case of functional genomics. So, in all these conditions, whether it is non-coding RNA, whether it is microRNA, whether it is protein coding RNA, one of the fundamental tools people use to measure and quantify the RNA is the age old concept, that is, your RNA can form a duplex if you have a complementary sequence. It could be RNA, it could be a single stranded DNA, but it does form a duplex. So, if you have a complementary sequence and if you are ways to measure whether RNA bound to that, then you are able to measure the RNA. So, this is the most easiest way of looking at it. And of course, you can always that if you have a double helix and you have the helix are held together via the hydrogen bonds that are found between the bases. So, when you destabilize them, for example, you can denature by, for example, exposing to the heat or salt concentration, then you are able to break the hydrogen bonds and then you can again allow them to form the duplex. So, if the duplex, one strand of course your RNA, the other strand is something that you are making for a given gene, and that strand has some probe, meaning ways to identify that. For example, you can add a fluorescent tag or you can put an isotope that is shown by the star here, then you are able to visualize that. Okay. So, this is the concept people have used since long to identify and understand, quantify RNA. So, how do you do? So, let us look into the classic approach. It is not dramatically changed. This is the first thing that you do. You isolate the RNA, the total RNA from any species. It could be human cells or animal or plant or whatever. Then what you see here, the two bands, these are separated in a gel. So, I am sure all of you know that the RNA can be separated according to the molecular weight. If the gel is run this way, the one on the top are larger species, longer RNA, the one that is below is a shorter species, shorter RNA. And when you put them across electric field in agarose gel, the RNA is negatively charged, it is going to migrate towards the positive pole, and smaller the RNA, faster they can migrate because you have pores in the agarose. So, this way you are able to size separate. So, what you see here, the two bands that are on the top is the ribosomal RNAs. What you see here is the transfer RNA, and what you see in between and you cannot see above is the messenger RNA, because they vary in size, whereas tRNA, ribosomal RNA, the large and small subunit have different size of RNA. There are multiple copies, thousands of them. So, you are able to see like a band, but the rest of the RNA which represent them, the messenger RNA is much less, it is 2%, 4%, 5%, therefore, and they are spread out different molecular weight. So, you see some of them here in between. If you apply more of the RNA, you will be able to see, but you do not see here is the microRNA others, which are very small, it would have ran out of the gel. So, you will not see. So, you need a different approach for that. So, this is a classic approach, you separate them in a gel, and then what do you do? Separate it by size, and then you basically transfer the RNA that are there in the gel to a membrane like what is shown here. You have the agarose gel here, and then you have the membrane, and then you have this paper towels which are nothing but which has a capillary power. It just pulls the salt solution from here. As the salt solution pass through the gel and the membrane, it carries the RNA with that, but it cannot the RNA cannot go beyond the membrane, it is stuck there, whereas you are able to transfer that. That is the way you are able to get the RNA, separated RNA on the membrane, and then you can put this membrane in a bag and use what is called as labeled probes. These are nothing but a short stretch of sequences representing the gene of your interest. Say I am interested in gene X, and I am taking a sequence, single stranded sequence of that gene X, which is complementary to the RNA, right. So, this is what we said, all the approach that we are using is the duplex, the ability of the RNA to form a duplex. Now, here we have labeled them because that is the way you can detect where the hybrid is, where the bound RNA is present. So, for example, you can label it with radioactive nucleotide, and then they go and bind because they form the perfect complementary sequence, hydrogen bonding, and then if you wash it, so all those that are not bound are removed. Wherever it is bound, you are able to see if you put an X-ray film, because then you have a radioactivity that reduces, it gives you that dark color, and the other region where you do not have the radioactivity, it looks clean. So, this way we are able to tell, one, whether the RNA is expressed, two, the intensity of the band, whether it is very dark, light, moderate, that tells you how much RNA, relatively speaking, is expressed. For example, here you can see that the RNA level in all three samples are same, right. If this was much more intense than the other, then you would say it is much higher as compared to sample 1 and sample 2. This is the way traditionally people used to quantify and measure and say whether RNA is expressed or not. So, this is called as the northern blotting, because it was evolved after the first method for measuring or detecting the DNA, which is done by southern blotting, wherein you separate the DNA the same way, run in a gel and transfer and hybridize, but here it was RNA, therefore, it is called as southern, it is called as northern. The first one was southern because that was developed by a person whose second name was southern, and since that denotes the DNA transfer, since the RNA transfer is different slightly, so this is called as the northern blotting. So, it has advantage because simpler, anybody can do it, but as with any other approach, it has its own disadvantage. One, only one gene can be analyzed at a time. You have a blot, you can detect the expression of only one gene. So, after that again you have to make a blot and look for another, and requires large amount of RNA, because you have to load high amount of RNA. So, if your sample size is small, if you cannot get that much of RNA, it becomes limiting factor. It is time consuming, it takes two days to complete the process just to know one gene whether it is expressed or not expressed. So, there are approaches people have used, and we will look into some of them. So, one of them is to go for an approach using a DNA. So, what do? You can extract the RNA and use the same principle, that is that you can have a small stretch of DNA sequence, single stranded, complementary to your RNA, and which goes and forms an hybridization like this. So, most of your messenger RNAs have poly A tail. So, you have taken a tissue, in this case for example the brain, isolated the RNA, and then you have selected the messenger RNAs, and you have used this particular sequence, the A poly A tail. You have used an oligo which is complementary to this, because you simply make multiples of T's, it go and bind, and then you use a polymerase called reverse transcriptase, the one that we described some time back. So, which copies the RNA into DNA, okay, it has become single stranded, and then there are ways by which we can convert the single stranded DNA into a double stranded DNA. So, basically now you are going to generate DNA which represent the RNA. So, if you have had more copies of that particular RNA, you are going to have more copies of that particular cDNA. So, it is not gene specific, it is going to do it for every RNA that is present there, which has this A. So, in other words, this would represent pretty much what you have otherwise in the RNA. So, what do you do with this double stranded cDNA? One can go and make libraries, that is what you call as cDNA libraries, but it also can be used for some other approach. First, let us look into the cDNA library. So, you have these RNAs which converted to cDNA, which is double stranded. Now, we can put them into vectors. These vectors, as I said, it could be plasmid, it could be phage, whatever it is, but we can put these segments inside, and these are made into cDNA libraries. So, you have each one representing one particular transcript and stored in a library. As and when you require, you can take them and look into that. In fact, people have used this cDNA library to sort of increase your throughput, meaning you want to look at expression of more than one gene, but using very similar concept that you used otherwise for northern blotting, how we can do. So, this is one approach, earliest approach, which led to what you call nowadays as a microarray. We will come to that a little later. What this particular group, this paper was published in 1982, what they have done is they have selected genes or rather the cDNA of genes, right, which they were interested, and then they took this DNA, denatured it, therefore it becomes single stranded, and then spotted on a membrane. So, basically what you are seeing here is nothing but a membrane. So, these are again the same nitrocellulose or membrane, right. So, what you do is I have say suppose 20 genes I have looked at, I want to look at their expression profile, then what I am doing is I denatured, made into a single stranded form, I am spotting it like this. Okay. So, this is the way I spot it. Each spot represents a given gene, and all you have is a single stranded complementary sequence of the RNA that you expect that to be expressing. So, what do you do? Now, you have the cells that has got the transcription going on. One way people earlier done is that in the transcription process you have added a labeled nucleotide. So, when the RNA is being made, it has an isotope added to that, right. Or there are other ways we can convert that into a RNA into cDNA. In that process you can incorporate, for example, a labeled nucleotide. So, this is the way to label the RNA or the cDNA that you make out of the RNA. Then you hybridize that to the membrane. Say suppose this is a tumor which is growing, this is a normal tissue, for example colon cancer I am looking at, the normal, for example, right. So, then you have the RNA, you have the same blot, same way I have a membrane in which I spotted the same number of genes, same amount of our DNA, and then I am hybridizing with an RNA which is labeled. Now, it will tell if I have a given gene expressed in high amount, and therefore, you would have more amount of that RNA, and that RNA being labeled, and if I have copies of that particular cDNA present on the membrane, it will go and hybridize. So, if I put an extra film, I may get something like this. These are the dots representing a given gene whose expression level is very high in this particular tissue. For example, colon, you have this gene expressing very highly as compared to, for example, these genes, there you do not have much signal. Whereas here you have certain genes that are expressed very highly in tumor, and some of them are expressed here as well, but some of them which are expressed in colon are not expressed here. So, this is the way you are able to tell in more than one, not than you are looking at one gene, here you can go up to 20 genes, 30 genes, we are able to spot them and tell the relative difference between the expression levels of the genes. So, this is the very first approach people have used, and then they went on to make much more densely spotted DNA onto a membrane using machines. Now, they have gone into machine because you can use machines to spot tiny amount of the DNA on the membrane, and this become commercial. There were in fact companies that were selling in 80s and early 90s, they used to sell this kind of membrane spotted with 20,000 genes or 10,000 genes or whatever it is. Now, that people have used to understand the expression analysis. So, that is what you see here. This is nothing but a membrane, and each spot represent a given gene, little bit of that complementary sequence, and then you have to of course isolate the RNA, label that, and hybridize to the DNA spotted on the membrane, and you can see that there are some spots that are coming up that shows that these are the genes that are expressing in a given tissue, and the level and so on. So, in this you can put it over a grid and you know what gene it is, right. So, this is the way they came, and in fact these there are about, they could go up to 4,000 human cDNA they are able to spot. It's a it was a huge shift from the way people are using northern blot. Now people are looking at one gene at a time. Now from hundreds you have gone up to 4,000 genes. That was a huge shift by looking at this kind of approach. So, this basically led to the next generation of approach which we now call as a microarray, right. Let us see what they are. The challenge now is that you have the cDNA clones, right, and the cDNA clones would have different size. Some cDNA would be shorter, some cDNA would be longer. So, when you want to really make an array like this, ideally you would like to have two things. One, you would like to have a range of the DNA, the size of the DNA, maybe 100 bases, 150 bases, not beyond, and so on. Two, you want to keep the base composition to certain within certain limits. The reason being, anything you hybridize, you have to get rid of the hybrids that are not completely hybrids, meaning there are some sequence matching, some sequence are not matching. That shows that these are not the perfect duplex, not representing the gene that you are looking at it. So, how would you get rid of that? You have to wash, meaning you have to expose them to certain temperature, which would destabilize any duplex that is not perfect. Okay? So, that you can do, this temperature you can regulate only if you have allowed a duplex of base composition all can retain its complementarity in a particular window of temperature. It all depends on how many G's are there, how many A's are there, right, because it gives you that strength. So, to all these things you should have some control over which region of the gene you are spotting, to what is the length you are spotting. So, these two cannot be done with cDNA clones, because these are randomly made, some are larger, some are smaller. So, it was very difficult to really control them. So, that is where they came up, that is the time the PCR had really come, something that most of you I am sure are aware of. This is nothing but you have a DNA strand which you can make multiple copies of it by providing a pair of primers. These are nothing but short oligonucleotides complementary to a given segment of the DNA, it goes and binds, and then you have the so called DNA polymerase. Here it is something which can function at higher temperature, not get destabilized by higher temperature. That is why it is called as polymerase chain reaction, because repeatedly you are going to heat, melt the DNA and then allow the primer to come and anneal and then make copies of it. So, this cycle you are going to make a short segment of the DNA. So, this by designing primers for a variety of cDNA, you are able to achieve two things. One, the size of the cDNA that you are amplifying using PCR approach, you have a control. Two, even the base composition you will have control, because you can choose a region that you want to make multiple copies. This is the way the PCR was done, but if you, instead of a DNA, if you use a cDNA in your PCR, then it is called as reverse transcription polymerase chain reaction, or RT-PCR. So, you have the RNA, you have a primer which makes a copy of that RNA into what is called as a copied DNA or cDNA, and then of course you can make it double, right. So, by using this as a template in a PCR, now instead of amplifying the genomic DNA, you are amplifying the cDNA which represent the messenger RNA, for example. Now, what they have done is the following. So, they have used these cDNA clones representing different genes using primers. They are able to make multiple copies of the cDNA and then make what is called as multi-well plates to make what is called as the arrays. That is what which led to the formation of microarrays. So, these microarrays, you call it a micro because it has small spots which is microscopic, array because it has got multiple spots. Okay? And what it helps is that in a small membrane or a slide, you are able to spot DNA representing thousands of genes. So, this cannot be done by hand like the previous one we have seen. People have tried to sort of do it by hand or to some extent automation, but this was much more challenging. So, they use the machines like what is shown here, automated robotic machines have come which are able to spot. And people have also used what is called as bubble jet. You must have seen bubble jet printers. These are nothing but printers that have a spray kind of a technique. It just sprays that color in a given region, therefore you can get the image just like what you have. So, they have used even that kind of a approach, and people have used even machines that used for the electronic circuits. You must have seen, if you open a computer you will find the circuits inside. These are nothing but these are printed on a chip. Therefore, it is also called as GeneChip, because what is being printed is a DNA over a chip using a very similar approach, therefore, it is called as GeneChip. So that is how, these are some of the machines to show that how the DNA was spotted on a glass slide, and then you came up with what is called the high density arrays, on a microscopic slides. You are able to now spot thousands of them, and that really led to what is called as a chip having thousands of genes, and now we are looking at expression of 10,000 genes, 20,000 genes representing the entire genome. So these are some of those setups people have used to spot the array. Now you see that it is going more like an industry. As I told you, genomics is something like more like an industry because less of a human interference. Most of them are taken care by the machines, and then just showing you some of the first or second generation chips that have come. This is from a company called Affymetrix, and what is shown here is a matchsticks just to show you the comparison, that you have the chip, each one having tons and thousands of genes spotted over here, this region they are spotted. Okay. So they represent, if for example if you take 2 chip, that pretty much covers the entire gene that are present, or at least protein coding genes that are present in the human or mouse, depending on the species you can take it. So what is done? So again these are nothing but the same concept, that you have a DNA single stranded onto which something comes and binds, which forms a duplex, and you are exploiting the base pairing, the hydrogen bonds between the complementary sequence. So how these DNAs are spotted? Basically you have fixed the one end of the DNA, therefore the other, the DNA is open for forming the complementary sequence. So these are fixed. So each spot, now you have multiple copies of the DNA representing a gene. So you will have thousands of spots, each one representing the DNA, right. So you have now the complementary sequence, which possibly represent your RNA or DNA, we will discuss a little later, which are again labeled, meaning you have incorporated some label by which you are able to now measure whether there is any duplex, right. So now such labeled complementary sequence will come and bind if there are perfect homology, right. So because you are going to pass them through certain temperature, therefore only the perfectly paired complex will stay there, others will be washed out, and now you are getting any signal from this spot, that means the RNA is bound there. How much is the signal? That will tell you how many copies are present. So this is the way you are able to measure whether a given gene is expressed and what is the expression level.